All right, so as we begin, there are some accessories to skin. Skin by itself alone is the major component of the integumentary system, but typically it also has what we refer to as just accessory organs or accessory components, accessory structures to the skin. And some of those are going to be nails, hairs, and glands. All of them belong to the integumentary system. And so your journey into this first organ system in lab is to really pay attention to this structure, which is a slice of skin cake. I know it sounds weird to say, but that's essentially what it is. It's that it's layering, or you can see that a section was made, or this little cube out of your skin, or a skin was diagrammed. And at the very top on the surface, you are seeing the superficial aspect of the skin. And if you were a needle, you would be going down into this direction, right? If you wanted to place an injection, right, and reach those blood vessels, or if you're putting a tattoo, that ink from your tattoo has to reach these areas. In last week, we learned about a tissue called stratified squamous epithelia. And in this case, we are seeing it here. We are seeing our stratified squamous keratinized epithelia, right? These cells is just going to be the layer up top. And we learned sort of a basic formula around this, is that when you have epithelial tissue, right underneath, you're going to have a membrane referred to as a, what is it called? Basement membrane. And so up here, this is a tissue that you should already be familiar with. And again, that's if you're keeping up. But if you're not, you should know, make sure to log away today, that it is your stratified squamous epithelia, but it's keratinized, meaning that it's going to form a layering or a texture that's like this. These cells, fundamentally, they divide. Their cells are a population of cells, of the epithelial cells called stem cells. And these stem cells are always going to be generating clones of themselves and sending that clone off to be a little skin cell. So they get to stay down here, they create a copy of themselves, and then they send that little cell all the way to the surface, and it becomes a little surface brick for us. And so when you rub off, or, like off a surface, or you take a shower, that little cell goes away and disappears into the drain. But these cells keep producing more and more and more. And so I would really like for you to remember that little bit of a story because, and I always would say the tattoo example as well, because the dermis is going to be the layer underneath the skin. This layer up top, the stratified squamous epithelia, is what we refer to as the epidermis of the skin. Everything above that basement membrane. And the dermis is going to be the side with all the connective tissues. On Monday, we saw that blood was a connective tissue. And what do we have on our slice of cake here? Blood vessels. So we are seeing one connective tissue in here, inside of these this blue and this red vessel. We are seeing some muscle here. We are seeing a smooth muscle that's connected to your hairs. And these muscles are there to give you goosebumps. And, in human beings, it's kind of a vestigial function, meaning that it's not something that we really need anymore. But, you've all seen a dog that, you're roughhousing with. And what do when they get a little bit frustrated or a little bit angry? They scruff up, right? And that scruffing makes the competitors, whether male or female, depending on the animal you're talking about, makes them look bigger. And also when they scruff up, like birds too, they can either sort of air themselves to cool, or they can relax and sort of tighten up all those hairs. And so they have their own mechanism for staying cool or warm in the outside environment using these skeletal muscles. But in human beings, we kind of don't really rely on fur as much anymore, other than for, fun. And so when we look at now these segments at the very bottom, we still haven't even given names to any of this, but you can see how it's all relatable if you've just sort of been keeping up with the class, that this yellow portion here is going to be your fat cells. And what's another name for fat cells? Adipocytes. So this is going to be adipose tissue. And then we have another family of tissues. Remember that if someone were to actually poke you, like, with a pencil, something sharp or something flat, you feel it. You feel it, period. And so you can see these black lines here. These are going to be representative of nerves. And you can even see there's even a nerve linked to your hairs. That's why plucking hairs, right, it hurts. And that's because your hairs are linked to nervous tissue. And overall, this is all your tissues that you've been learning about, all seen in one organ system. So now we're just going to get very specific as to which ones you need to know and what we need to know for our exam. So most diagrams in the lab, we're going to see the models. They're on your tables now. Some version of it is going to be look like this, like a layer cake. You're only going to be seeing one side of it, really. And you're typically going to have to sort of start seeing how small the epithelial layer is compared to the connective tissue layer. That's one of the first things you note. Those little tiny layers might have letters at the very end of the model, and those will be representative of something we're going to talk about in a second. But otherwise, you want to make note of how small this is, but believe it or not, there are 5 layers you have to know just in this segment. So keep that in mind, that as you explore the epidermis, there are 5 layers in that epidermis that matter to us. 5 layers. And then underneath, we're going to encounter all those tissues that we discussed on Monday for the most part. And so once we hit that basement membrane of the epidermis is made out of epithelial tissue. The dermis, underneath the epidermis, is made of 2 layers. One is referred to as the papillary layer. The other one is referred to as the reticular layer, with the papillary layer being the smallest one. And so it might highlight here, this bracket is sort of just this segment. And the good news is this is just a unique name for that first layer right above these unique vessels, which is not on your terms list, so I really don't want to actually add anything that I don't need. But what I want you to draw your eyes to is that in that papillary layer, there's a boundary, and the good news is it's made out of 2 connective tissues that you know. It is made out of areolar tissue, just this segment here, areolar tissue in the papillary layer. And remember, if we just quickly look at our histology, I'm going to quickly use this because I just have quick photos for these tissues. They're the same ones I use on your other slides. I just sort of been working on these. And so when you look at this, areolar tissue had fibroblasts, and they produced fibers. Remember that? They were called fibroblasts. These large pink ones were collagen fibers. The thin purple ones were called elastic fibers. And this tissue specializes in allowing white blood cells to sort of swim around in these empty spaces to navigate for dangerous things. And rightfully so for skin, right? Because if you get a cut on a scrape here, you need sort of a tissue that allows white blood cells to be wandering around this little segment. Sometimes you'll even see in textbooks that they refer to those white blood cells as wandering cells. And so in the papillary layer, you encounter areolar tissue. That's why on your list, you may see that the dermis has a papillary layer, and then you'll see that it says areolar tissue underneath it. And then next, as we go more deeper into the tissue, we'll see reticular layer. And now reticular layer, just this segment still, is going to be an area where a lot of things are deeply rooted, such as the majority of the glands. We're also going to see hair follicles, or the origin of hair, where they sort of embed themselves and grab all the useful nutrients, right, through the blood vessels in order to supply themselves with all the protein that they need to make themselves grow. And then you have glands that maintain hair. And so these areas contain highly specialized structures for a lot of these structures we're going to talk about today. And so, but reticular layer overall is made out of something that we already explored on Monday called irregular connective tissue. And just to have a complete little review here, as we explore a picture of dense regular, this is the picture I think all notes typically use, looks like this. And then you will see you have your fibroblast, and they are using collagen in a different manner than areolar, that their job is to be a lot more firmer. And so this was dense regular, pardon. And so when we go to irregular, yours doesn't have it, sorry. I should just use yours, 1 second. I have it on this one. I did not put it on your iPad, so if you're watching this recording, I'm showing your chapter 3 and 4 tissue slides, and I'm just going to the page with irregular on it. And so when we look at irregular, Irregular was different from dense connective tissue because dense had used collagen in a very wavy, parallel manner, and this was very tough. Remember, it serves tendons and ligaments. But when we examine irregular connective tissue, sort of related to regular connective tissue, these cells lay out collagen in an irregular manner, so it doesn't have the type of strength that dense regular has to hold bones into muscle and bones to other bones, but it has the strength and the flexibility that you need underneath your skin, because this is the reason that when you pull your skin, that it doesn't just rip off. And then, you have to go grow brand new skin. And you can see here in deep portion of the skin or the dermis, this is the best example for the location. And this even tells you what the function is, because if it's underneath the skin in the dermis, then you can assume that it's to allow for some stretchiness and some toughness, right? We really don't want it ripping off. And so with that, back to my iPad here. And so this whole reticular layer contains, again, irregular collagen fibers all over the place laid out by fibroblasts. So you can see that the fibroblasts in this area and the fibroblasts in this location secrete collagen in unique and different ways. This one's creating areolar tissue. This one's creating irregular tissue in the reticular layer. So we're sort of building our cake, right, with tissues that we're slowly learning throughout the week. And you can see that in this dermal layer, we refer to the dermis overall as something called being vascular, implying that it has blood vessels to bring nutrients to it. And so epithelial tissue generally has always been considered an avascular tissue, because you'll notice that there's no blood vessels that actually make their way all the way to the very top here. That's why it was very important for me to regurgitate over and over to all of you that, oh, what's the layer after the basement membrane? Connective tissue. So that to drive home the point that this is uniting, right, all the tissues. This is uniting your digestive tract with your epithelium, your skin, because it'll drop off the nutrients, and the nutrients will be used by these cells to make more cells. It'll be used by the fibroblasts to make more collagen, more elastic fibers. It'll be used by these fibroblasts to use collagen in an irregular manner. And so you can see why blood is considered a connective tissue. It is bringing nutrients to everything, and epithelial is always typically considered avascular because it relies on the connective tissue below to receive all its nutrients. And so, yes, that's sort of a big take-home in lectures typically. And then lastly, you may hear about hypodermis, but just as an FYI, it's not even considered a layer of the skin. Epidermis and dermis are considered the skin boundaries, the skin layers. But right underneath this region shares a very similar name, but it's actually not considered one of the layers of skin, even though it's typically called hypodermis, and sometimes you'll see subcutaneous layer. And so in this layer exists, again, you see this pink here, this is going to be areolar tissue once more. You have areolar tissue, so you can see how all this builds up, areolar tissue up here, then you have irregular tissue down here, and then you have areolar tissue here, and do not forget our adipose, right next to our little fibroblast here, making areolar tissue. So our adipocytes take residency with them. And so you can see that all organs can be analyzed in this manner. How does it get blood? How does it get nerves? Are there glands? Is there fats underneath it, above it? And just kind of, you typically see these same trends throughout this entire semester. And so with that, some things to introduce is that, remember that I say hair, nails, and exocrine glands, they are typically referred to as accessory structures, and they're going to include some of these glands. There's going to be different ones. They may look the same sometimes, but they may not be the same one. So we're going to make sure we explore those today as well. And so as we move forward, this is just a regurgitation or a repetition of what I said about papillary layer. It contains our areolar tissue, and it's going to contain the area for capillaries. We may not know what lymphatic vessels are, but they're just vessels to help regulate volume and prevent infection. And then, of course, we have nervous tissue so that you can feel things, right? And then something to draw your eyes to, which I believe is not coming up next. It comes up in a little bit. is that you'll see that the nerves here are going to play a different role, or the role they play really depends on how deep they are. If they are more superficial, they're going to play a bigger role in paying attention to details in touch. Meaning that if I put quarters and I put nickels and dimes and pennies and even foreign coins in your pockets, you could all probably dig around with your pockets with your fingers, like, you know, not looking at it, and you could probably pick out a penny, probably pick out your quarters, your dimes, because you have very sensitive or nerves that are very important in just detecting very tactability, meaning precise touch, that you can sense the difference in texture because of very fine touch. And then there are others that are located deep within the dermis that are going to be responsible for deep pressure. And doesn't that make sense that only when someone's really pushing you downward, right, that you're pushing you, the skin, and all these collagen fibers, all of these cells, everything's being pushed down, and then finally these detect it because they got pushed against by all the material that's there, all the protein fibers. And so it's pushing down, and then these are responsible for communicating to the brain, hey, someone's really pushing you pretty hard. This is uncomfortable. And so that's kind of why you see them in different locations, as we're going to see in a bit. And so with that, anything else to add? I believe we can move on from here. And so in the lab, we'll also see some little models that have nails, and nails are considered part of the same system. And so these nails, as you're seeing here, is going to have some boundaries to it. The boundary that's going to be the tip is going to be referred to as the free edge. The area that's going to be the inferior portion is going to have a little bit of layer, a little flap of skin that some of individuals that may go get what is it called? Pedicure. When they go get a pedicure and then it gets pushed down to the very bottom, right? They remove that excess skin. Even as you look at your nails now, that little bit of skin is called your eponychium. Eponychium. We call it commonly the cuticle. The cuticle. You can call it both in our class or for the exam. And then finally, you'll see that the nail itself has a pigmentation, or a very light color, pardon to mean to say, an absence of pigmentation really, and it's called the lanula. Lanula. And just as you look at the nail and you look at are stratified squamous, and we're going to explore this a little bit more in detail again in a little bit. But remember that the story generally goes that there are cells down here that are dividing, and then these cells are programmed to fill themselves up with a protein, die, and become little bricks. But the source of the cells is always going to be at the base. And so the lanula represents, or is actually important for that reason, because there's something you're going to see in a little bit called stratum basal, and don't worry about it right now, but it just means the layer underneath. And so this is going to be generally the site where the nails are kind of growing outwards, just like the skin. And it turns out that nails are actually using the same protein that your skin cells are using to make that skin texture. And so that's sort of why the nails are related to skin. But in our lab, the only thing that's important for you to know is that the overall gross anatomy, free edge and the lanula. Finally, you have your nail fold. That's going to be the sides, the lateral sides of the nail, where you see this sort of indentation. I know, I wish this little arrow was a little bit better, but this should be on the groove here of where your nail sort of really ends. Oh, it's right here. Sorry, nail groove. Sorry, nail groove. And then nail fold is going to be fold over. It's going to be where we rotate over. Pardon. Trip myself up right now. And so, again, nail groove is going to be the site of attachment. Site of attachment. And then the fold over is going to lead to the skin over. Yes. And yes, let's keep it going. And I'm actually curious as to why I'm not seeing a very useful slide that should have been in the front. And I just want to make sure that I address it. Yeah, I feel like I'm missing a slide. Okay, well, I'll pull it up right now and then we'll talk about that segment. So let's finish up the nail. And so as we look at the nail and you make a sagittal section, we have only one model that does this very image. And here you can see the bone. And what connective tissue makes up this then? It's a very simple answer here. What's the name of the connective tissue that makes up our bones? That's the name of the cells. The connective tissue is just called bones. So the bones here, that's just bone. That's the connective tissue. And what are the cells? I heard it back there. Osteocytes. What are these little yellow globes that I keep saying? Yellow globes represent fat cells. So what are these? Adipose. And then here on the skin, what's the epithelial tissue at the top? Stratified squamous. And so, let's keep it going. And so as we see our nail bed, like when a bed has a body, right? If you look at your nail, the very skin underneath the nail, like you've all maybe have lost a nail in your life by accident, and then that very sensitive part underneath it is considered the nail bed. That's going to be made out of still skin material, right? Skin. And then the nails sits right on top. And overall, let me see what other ones you need, because you don't need all of these. And then nail root, just like the root of a plant, that's going to be the segment that embeds itself deeply, going in a proximal direction, right? Not going towards the fingertip, but going towards the upper shoulder, going towards the trunk of the body. So the nail root embeds itself. Here you can see the lanula again from the side, so it's a little bit underneath. So if you look at it with this image, that nail root is sitting somewhere deep within this skin here, underneath that. And so that would be our nail root. Now these words are probably going to be a little bit easier to see when we look at a microscope in a second. And there's just one last word to touch on, and it's hyponychium. And so it's going to sound a lot like the word eponychium, just like this one. But that's the skin on top of the nail and at the bottom. And here hypo is going to be underneath. And so when we look at hyponychium, it's going to be the skin right underneath that fingertip. Right underneath. And so let's look at our hair. And so our hairs and nails share a little bit of a similar terms. And before I actually discuss the microscope image, is that when you look at these structures around a hair bulb, if you look at the hair itself, at the very bottom of each hair will always be a rounded bulb, like a light bulb. That is what's referred to as a light bulb. And since I'm using this as a PDF right now, the PowerPoint itself will put the arrows for you in order of the bolded terms. But right now, since I'm using this, I'll just have to do it myself. But a hair bulb is going to be the ones that are found at the very bottom, right, the bulb-like shape. Hair root is going to be the actual hair on the inside, not the actual bulb itself, but the hair inside, the hair root. And then the hair shaft is going to be essentially the region that goes out of this. So you can consider all of this the hair root, to keep it simple and also more accurate. Hair root is everything underneath the skin, right? Everything that's embedded into the skin. Just like the root of a plant is embedded in the ground, the hair is embedded into your skin. It's a hair root when it's in your skin, and it's a hair shaft once it's out of your skin. That's the difference between these terms. And the last thing, the word follicle. The word follicle is the same thing or the same concept as when you think about, like, you know, you all see movies of like samurais or some cartoons or something, and then they always have a sheath in their sword, right? They have a sword and they put it in a sheath, right? So they don't cut themselves as they're walking around. That's what a hair has. Every single hair will always be within a container, a sheath. That sheath is called a follicle. And so that follicle typically will wrap around the entire hair. So I noticed in this slide, here's my pointer, that this one on this model, the follicle is completely full, right? You can see the outside of the follicle and the hair is on the inside, but on all these other ones, they cut out the follicle or half of it, they make a sagittal section of it so that you can see the inside of the hair. But otherwise, it should look like a complete sheath around the hair. If the follicle on your model is intact, it will look, you won't be able to see the inside hair, just like you are seeing on all of these. And so again, a follicle is just generally like a sheath of a sword, something that wraps around your hair. When you look very closely at a hair bulb, right, we're looking at the bottom of those hairs. This is essentially where all the nutrient absorption and capturing is occurring, or utilization is occurring, because what we see is that when you analyze the entire bulb, inside you see something referred to as the hair papilla. This hair papilla is going to be like probably a little bit of areolar tissue, and it's going to have simply blood vessels. Because every single one of your hairs needs a supply of proteins to make itself. Even like they say a lot of times, like, oh, my hair looks so much better when I'm on my diet's better. And it's probably because you're consuming more protein, because your hair is made out of protein. And so that's why typically when you're consuming protein, you look like you have healthier hair. And that's because this region here is directly tied to capillaries that will supply all the nutrients so that all these cells can continue to divide throughout your lifetime and continue to grow your hair. And so within that area, the matrix, this is where all the hair cells are. They're grabbing all the nutrients from the blood vessels in this area. And so all those blood vessels release those useful nutrients, and then the cells that are here are just going to be doing exactly what the skin cells do. They're just going to be undergoing mitosis. They're just going to be making clones of themselves. They're just going to make copies of themselves. And those cells are destined to just become dead little hair cells that eventually probably get washed off or cut off in our daily lives, right? And so otherwise, all you need to know is the bulb around. You just need to know hair matrix is going to be the area around that papilla, sort of the region I just highlighted. You'll even note on this microscopic image that it is a little more deeper purple, more violet compared to this region's out here. And then finally, hair papilla is just this region in the center. That's where the blood vessels and the nerves are. This is why when you pluck a hair, it hurts. It's because they link to your nerves right there. Your brain connects to all your hairs. Pretty cool, right? And in some animals, like that have whiskers or rely on whiskers, their nerves, they use them sort of like fingertips. Like, you ever wondered why dogs and like cats typically rub against things? They use their whiskers, cats a little bit more specifically, will use them to sort of navigate around, to not touch things and navigate a little bit better in the dark, to feel for things and dogs will feel for things in the ground a little bit better with them. So they use them like little fingertips because they have more delicate or more sensitive nerves, and they can draw more information from touching it as opposed to us, right? If you want to feel the texture of this, I'm not going to use my head, and I'm not going to know what the texture is, right? It doesn't make sense. But they can do that with their whiskers. That's why they're a little bit of a different texture, and just a little unique factoid about them. That's why you'll see rodents have them too, right? They need to navigate in the dark more and things like that. And so on our models, where it matters, this is going to be a nice little view of a blown up image. It's kind of very tiny model. It's kind of like comedically small. But I set it up in the back, because in the back I set up a practice exam for all of you, so you can see the style of questions for this upcoming exam. And you'll be able to play with it in a little bit. So it's back there if I'm not mistaken. Or maybe I moved it for something else. But if anything, I'll bring it to the front. And you should be able to see exactly what looks in their cartoon image. You have the follicle around. You have the bulb. And then you have the little region right here, which is going to be the little papilla. Right? And the matrix, I probably wouldn't ask you on this one. It's just too hard. And then you can see it on this one, though, that they actually take out a little hair. And then you can see that little papilla. Oops, sorry. The little papilla is right here. And then the matrix is right here. And these cells are just dividing. And we have to talk about that too, that little dividing in a second. But otherwise, that's going to be our matrix. And that's really it. And then there will be like a little fingernail that's cut in this manner. So just know the basic nail root, lanula at the very top, the nail bed, the hyponychium, eponychium, or cuticle, and that's all. And so the ones that are on your list. And then do not forget that you're going to see a muscle, and this is a perfect example of another muscle that you learned about already, smooth muscle, which is involuntary, and it's attached to the hair, and it's referred to as erector pili. That's the name of this smooth muscle. That's the name of this one. Right? There's smooth muscle all over the body, so you got to give it a name. Otherwise, you don't know where it's smooth muscle of a gland, smooth muscle in your intestines, smooth muscle in various places. And so with that, let's move on. And then we have our glands. Now, a little fun fact about our glands, and we can review a little bit more about tissues from last week, is epithelial cells. We learned the tissue cuboidal cells, right? We either have simple cuboidal or stratified cuboidal, but otherwise simple cuboidal, the one that I care about more than anything, I'll probably take off stratified cuboidal and stratified columnar. But otherwise simple cuboidal, you always know you're looking at it because you're seeing cube-like cells, as close as you're going to get to it, because the cytoskeleton of these cells make them cube-shaped. And so remember that we discussed that these are typically found in glands. And that's what your slides say. All they have is just a big word glands there. And again, they're just found in glands. And so these glands themselves, the ones that we're going to talk about right now in the model, there we go, that in the model, like this one, this one, this one, inside of them, they are all made of cuboidal cells. So now as we're really understanding how all of this is coming together, up here we have stratified squamous, keratinized, and then in here, we have cuboidal cells inside of each one of these ducts, tubes, and inside of this gland. All of it is mainly going to be cuboidal cells. They may more than likely be simple, or the larger one might be stratified cuboidal. But otherwise, it's not a fact I care too much about here in lecture. That might be something you will address. That's why I'm sort of taking my time to explain that. But otherwise, one thing that you do need to know in lecture, and it's worth mentioning in here, is that there are different types of secretions. We learned in our cell biology portion of our class that every cell has a nucleus, right? Each one of these has their own nucleus. They have their own Golgi body. A Golgi body is going to use those proteins and all the carbohydrates that were made by the ER, the endoplasmic reticulum, rough and smooth, and they're going to place it in the Golgi body, and the Golgi body is going to package those products. And then a cell, like a gland cell, can then secrete those products out of the cell through exocytosis. And so that's what these cells do, is that they can secrete out and then, secrete whatever they're creating onto the surface. This is referred to as merocrine secretion. And so an example of this is that we've been learning columnar cells typically can secrete mucus. And so this is how they do it. Even cuboidal cells can secrete mucus too, to an extent, depending on where we're at in the body. But otherwise, whatever needs to be secreted out of the cell is just done through regular cell biology, right? The Golgi body delivers things through the vesicles, secretes it out, merocrine. Now, when we look at apocrine, you were going to see this in lecture, guaranteed, a piece of. That's what you should use this word to help you remember how this gland works. Because these cuboidal cells are crazy or different, right? Their secretions, they place them in the Golgi body like before, but instead of allowing for secretions to go out of the cell, instead they pack it to the end of the cell. They pack it there. And then all of a sudden, they just completely pinch off that whole cytoplasmic part of it. They take off a complete chunk of themselves with all the stuff that's supposed to be in there, and then it goes out into the outside world. And then it regrows it and does it all over again. So this is referred to as an apocrine secretion. And that's when a piece of the cytoplasm comes off. And you should use that, a piece of cytoplasm. Rips, I don't know, you can keep going with it, but yeah. And an example of this is going to be in the mammary glands, right? And then lastly, we have holocrine gland. In Latin, holo means whole, right? Like the entire segment of something. And so think about that the cells themselves, they're going to die or end themselves in order to, provide their function. Here you can see cells are dividing. You are seeing the chromosomes being lined up. What phase are they in when the chromosomes are lined up down the middle? Metaphase. What is it when they're growing apart from the metaphase plate? Apart, anaphase. And so they're dividing and creating more cells that will eventually look like this. And then these cells will just build all the stuff inside of themselves and then just end their lives or end their existence and then explode and allow all those things to exit out. And so that's where the word holocrine comes from. So this is something you're probably more than likely going to see in lecture. And we're going to see an example of all of these today in the skin as well, in different places. So let's get into that next, finish up. Oh, and the last thing to add here, this is more for like a deeper understanding of these terms. When we talk about exocrine, talking about the skin is a perfect time to talk about exocrine, because in the skin, exocrine, the word itself just means like one of these glands, for example, one of these. When they secrete something, it just ended up right here on the surface of the skin. It just ended up right here. That's it. That's as complicated as exocrine glands are. I mean, it just means a gland that secretes right out of a little duct, out of a little tube, and places it. Like sweat, as an example. Sweat will just go out right out of the pore, and then bam, you're full of sweat. But what is endocrine now? You all have heard of the word endocrine. It's its own system, and it's also a word to describe a form of movement of molecules. That the only way that something becomes endocrine is that if a gland secretes something, and then it places it into the blood vessels, and then the blood vessels deliver it throughout the body. That's what an endocrine secretion is. An endocrine secretion places things into the blood vessels to be delivered somewhere else. An exocrine gland secretion is like me doing this. It's like, oh, this is where I need to put it. That's all. It's just like within distance, just right here. But endocrine needs to put it in the blood vessels. That's like if I mail this and I mail it to my house instead of taking it with me. So that's the difference between endocrine and exocrine secretions. And so mostly we're just talking about exocrine secretions today. So that's just a nice little introduction to those words. And then so we have holocrine glands, which are going to be glands that secrete oil in the skin. Holocrine. They break off entirely. In the lab, you just have to be able to ID these things. That's all. There's no function questions for anything today. It's just ID, ID the terms on your list. And then you'll have one more called sudoriferous. And how do you say sweat in Spanish? Anybody know? Sudor, or sudor. So this means sweat. And so this is sweat glands. Sudoriferous glands. That's where, in Spanish, this is where the word comes from. It comes from the Greeks, the Latin sudor. And so here, let's keep it going. And so as we keep going, we look at our sweat glands. That's the first one we're going to look at, the sudoriferous. You can call them sweat glands or sudoriferous. It's just in case you see either one. And when you look at them, they are a form of merocrine secretion. Or once more, the type of secretion that just does regular exocytosis. Just regular little vesicles exit the cell and secrete right out of the little cell. Nice and easy. Merocrine secretion. You may also hear them being called eccrine glands. And so that's just in case you hear it. But otherwise, merocrine just works just fine. And you find these in the forehead, palms, and on the soles of your feet. That's why when you get a little like nervous, right, you get a little clammy, it's the palms, right? Even some individuals, the soles as well. And then it secretes mainly water with electrolytes. If you're not sure what electrolytes are, it just means salts like sodium ion, chloride ion, potassium ion, things like that. And its role is to cool the body when it's hot. And then our next topic here is apocrine. Apocrine, a piece of cytoplasm. These glands, this is going to be the larger one on the model. Oh, sorry, I didn't even tell you which one was on the model. It's going to be the little tiny white ones that you see on these models. They're going to be smaller. And then you're going to see two levels, right? You're seeing one small and then one huge one. The small one is our merocrine gland or our sweat gland or our eccrine gland or our sudoriferous gland. Just pick whichever one you like best. And then the larger one is going to be apocrine. And this apocrine is going to produce a more smellier secretion. And so this particular gland is more apparent during puberty, and it's what creates a thick, cloudy, and it's going to be full of proteins and lipids that bacteria love. And so body odor, when someone smells bad, you know, they have BO, it's typically bacteria on the skin are consuming those oils, and they're growing. And so the bacteria, I don't know if any of you taken micro before, but bacteria, when they're concentrated, they stink. This is bacteria. And so that's what BO is, or bad body odor, is essentially bacteria consuming some of the things that you're secreting, such as proteins and lipids. And so with that in mind, you can see that during puberty, these become incredibly active in the armpits and the pubic region in young individuals, because that's their process, right, into growing into mature adults. And then we have other apocrine glands found in places like the ear. This is called ceruminous. And then this is going to be earwax. Your earwax itself is essentially like the oil that your armpits secrete, but they add a little bit special something to it to make it into earwax. But that you do not need to know this for the exam. It's just something that will come up probably later. And then another form of apocrine gland, as I mentioned, the mammary glands, that the most successful way for these cells to secrete that extra milk is to sort of pinch off a piece of cytoplasm, because they produce the milk products into the cell, and then the cell just cuts it off, and then all of those go into the gland, to the tunnels or the ducts that will lead out of the nipple, and then finally provide some milk for the young. And when we get to reproductive, we'll go over the specifics of that as well. And then we have our holocrine glands, another one that secretes oils, and again, the whole cell dies in this process. And it's going to be these bluer ones on your models. And these are typically going to be easy to identify because they're associated with your hair. And so all the glands that are found directly on top or attached to a hair follicle or hair in general is going to be referred to as a sebaceous gland. It's in the categories of holocrine because, again, the whole cell dies, but it's referred to as a sebaceous gland, these blue ones. Yes, and then so these are the ones that the reason that I think the ladies in the class are a little bit more familiar with like hair care, but otherwise, like, why don't we shower every single day? Or anybody can answer, not just the ladies.
Because we don't want to stink.
Yes, we don't want to stink. We don't want to stink. But as far as hair goes, hair maintenance, what does it do to hair if you take a shower too many times a day?
Oh, yeah.
It dries out, and what it's taking out or you're washing off is the secretions of the sebaceous glands. And this is what makes hair shiny and look healthy, is those sebaceous glands, and keeps it moist. And so that's why over washing or over showering can dry out your hair follicles and make hair look a little drier. It's typically because you're washing off very useless secretions. And I wish I could tell you, last semester I had someone who worked in a dermatology office, and she had all these fun facts about hair care that she shared with everybody, but I don't remember them now. Wish I would have written some of them down, but so I can share them in the class, but I don't remember. All right. And so with that, the last thing to show is that we've explored all these structures inside here now, and the only thing to touch on now is what does the stratified squamous epithelial layer look like? And so we're going to need some notes for the specific notes.